Hi, in this video we'll ask what kinds of biological questions you can address using Drosophila. We'll also talk about the broad strategies that are employed to understand the functioning of specific genes. Drosophila are independent, fully formed organisms, just like us. So questions that you have about the functioning of a living organism that can range from a variety of processes, some of which could be your thesis, would delve into the molecular mechanisms behind interorgan communication, inheritable human diseases, immune functions, tissue repair and homeostasis of any number of tissues, metabolic regulation in development and homeostasis, and fundamental gene transcript or protein regulation. You can also use them to study neuronal circuitry and behavior and the evolution of all of the above. The molecular mechanisms are important because they present therapeutic or intervention targets, and you can ask a lot of other questions as well. So there are broadly two ways of framing questions in Drosophila. You can either ask which genes are involved in a specific biological process of interest, or you can ask what does a specific gene do in a specific biological process, or you might just want to know what the gene does everywhere. So it is important to start with a well-defined question. You can use the methods available in Drosophila in a combination or refinement of these above questions. Now, the broad experimental strategy of finding out how any part of any system works is to remove it, see what goes wrong, put it back, and then show that with that part, the system comes back to the way it was. That says that that part was responsible for that thing that went wrong. So a loose analogy that one can use to a fly and these two questions that we just framed would be a car. So if I wanted to find out what a steering wheel does, I remove the steering wheel and see how does the car go differently. It starts, I can change gears, I can move it, but I cannot turn it, so it's possible that the steering wheel has something to do with steering. It's a very, very loose way of putting it, but this is to get you acquainted with what it means to be using Drosophila as a tool. So in terms of a car, we can ask, instead of what genes are involved in what process, I can say which parts of a car are involved in car ignition, or what does a spark plug do in car ignition. Very, very specific, right? Now, so for example, let's start with the first question, which parts are involved in car ignition? What you would do is to take a whole bunch of cars and do something to disrupt their function. Disrupting their function, it could range from something like an electromagnetic pulse, or it could be running over them with a tractor. The specificity and efficacy with which your method of disruption works is important. So in this population now, after having disrupted their function, let's ask which of these cars can no longer function. And so it turns out there's no ignition in these cars. And so now you take a look at all the parts and you find that the spark plug, the engine, and the fuel tank were disrupted in your initial perturbation. What that's saying is that these parts may be involved in car ignition. This might give you an insight into what the process is. Now with flies it's quite different, because you have some sense of the layout but not everything, which is why you're using them to discover new things. Let's ask the question, which genes are involved in body patterning? So I take a bunch of flies and then perform disruption, like I said, which in this case is likely to be some sort of mutagenesis. And then I look in their progeny, which there will be many, and from those thousands we find which individuals have a problem with body patterning, which of them have the segments of their body switched in order, or they lack specific segments. And maybe I'll find one progeny, with a body patterning defect. Now you have to find out what gene it exactly is. And then you have to map the gene physically onto the chromosome. The chromosomes are long DNA stretches, and you have to tell me exactly along the length of DNA in what sequence there has been a problem that is giving rise to this body pattern. Now let's reframe the question slightly with, what does a spark plug do in car ignition? So what I would do is to take a bunch of cars, not touch one part of them, and then half of them I can use as your test subjects, in which you take away the spark plug and see what goes wrong. You probably don't know what it might affect, and so you find out that the cars don't start, suggesting that the spark plug has something to do with ignition. The kinds of information you get depends on how you frame the question. So specifically, let's think about what does a gene of interest do in some favorite biological process. So something that I'm familiar with was the gene CTR9 in germline maintenance. So what you do is to start with a bunch of flies. In some of them, you do not cause any changes. Those are your controls. But in some of them, you do take away the functioning of CTR9, specifically in the germline. The germline is where your gametes come from, the egg and the sperm. So in the control sample, where nothing has been changed, you find that their ovaries are looking nice and healthy, and compared to them, the ovaries where there is no CTR9 are tiny and shriveled. This tells you that CTR9 is required for germline maintenance in Drosophila. Now comes to the challenging bit, which is to find out how. What that means is you have to find out where this protein CTR9 is located within a cell, what kinds of other proteins it interacts with. Does it cause some changes to a chemical reaction? Is this an enzyme? Does it affect the structure of the cell because it is sitting somewhere on the cell membrane, thereby acting like some sort of scaffold? Does it serve as a platform for other proteins to get together to form a complex and then affect an enzymatic reaction? What does it act on? Those are the kinds of details that you can get into. And then, of course, there is a recombination or refinement of the above questions. You basically have to ask, how do you non-specifically disrupt a gene's function? That is a question you have to ask. And it's important, like I said again, because if you run over your car with a tractor, that tells you nothing about the functioning of individual parts. All that you know is that a car only works if it is whole. So understanding the methods in which you can non-specifically target genes' function is important, and then what kind of disruption they cause is also important. Then you ask, how do you specifically disrupt a gene in a specific tissue? If I want to take out the spark plug, I need to know where it is, I need to know how to take it out, I need to know how to put it back. Figuring out the methods behind this is also important, and this is something that for which you must understand and wield Drosophila genetics well. So what does this entail? You must understand the number of chromosomes and how they are passed through generations and how to follow and select for them. I mean physically, the chromosomes that are lengths of DNA wrapped into each one of a fruit fly cells. You have to know which one is in which progeny. You can distinguish them using tools available in Drosophila. And then you have to ask the what, the how, and so what of mutations and mutagenesis. These methods you have to be in full grips with. How does your screwdriver work? Do you understand that? How does your drill work? How does your frying pan work in terms of some sort of disruption? You need to understand that. Another aspect of genetics is meiotic recombination. You have to know how to wield it, how to use it, either to bring together some sort of intervention or mutation that you have created with a known background intervention or mutation, or you have to be able to prevent that from happening because you've created a specific mutation, and meiosis can bring together mutations that you are not interested in at all, and that can confound your result. So you have to be able to understand how meiotic recombination works, how to use it, or how to prevent it. It is important to change the function of the gene of interest. You can either turn up its function using overexpression, or you can downregulate it or take away that gene entirely. How do you do that? In specific tissues, because sometimes if you take away a gene, the animal dies. That is useless information, especially if you're looking for an adult structure. So you have to be taking away that gene in that tissue of interest in adults. What are the methods? These are things that you need to learn, and that's what the series should help you with. And finally, you need to be able to use combinations of genome rearrangements, transposons, molecules from other species as tools for all of the above purposes. You need to know your flies, the notation, mutations, markers, recombinations and balancers, molecular tools like UAS Cal4, Lexay operator, Flip FRT, and some of their applications like in lineage tracing, generating null cells in a heterozygote, and so forth. And so that's what we'll do in the next part of this series. Again, if you have specific questions and comments, please reach out to thed2lab@gmail.com. And we're also on X at thed2lab and on Blue Sky at thed2lab.bsky.social. Thank you.